Now, the next talk is being presented by Alexey Gurevich from St. Petersburg State University in Russia. That's one of the proceedings presentations, and it's entitled NPS, Scoring and Evaluating the Statistical Significance of Peptidic Natural Product Spectrum Matches. Alexey, please.
Thank you for the introduction. Hello, everyone, and I am very happy to present our new work. And the title is pretty complex, and I will try to explain it in more details. First, about our target compounds. It's peptidic natural products, so simply PNPs. And the simplest definition is just small bioactive peptidic molecules, like this one. And if we would like to be more strict, it's two classes of peptides, non-ribosomal peptides and ribosomally synthesized peptides with post-translational modifications. And they are important because they are a rich source of new medicines, including antibiotics and other important agents. Like this one is penicillin. It's a non-ribosomal peptide. And we usually use mass spectrometry analysis to look for peptides and PNPs. And in very basics, it looks like this. We get a sample, process it with mass spectrometry instrument, and after that we can computationally compare this experimental mass spectra to chemical database or spectral library and get some identification, so a pair of chemical compound and a mass spectrum. And this problem is difficult, but it is almost solved in traditional proteomics case, where we talk about linear peptides. For example, there are a lot of tools for spectral library comparison and chemical database searches, and even for de novo peptide sequencing, and like the previous talk. But these tools are not applicable to PNPs because of completely different structure of these compounds. For example, PNPs are commonly non-linear. They are mostly cyclic or branch cyclic or even more complex compounds. Also, they contain non-standard amino acids and a lot of post-translational modifications, which also complicate their discovery. And current spectral libraries for PNPs are rather small comparing to proteomics spectral libraries. And until recently, there were virtually no tools related to mass spectrometry-based high-throughput identification of PNPs. But things are going to change in future. And a few years ago, University of California in San Diego started a large GNPS project. It's a huge platform for sharing and analyzing mass spectra related to natural products in general and peptidic natural products in part. And this is the number of users at 2016, and now it became larger and larger. And this platform both shares this data and has some tools for its analyzing. And there are at least two tools for in silico analysis, so for comparing experimental mass spectra to chemical databases using some computational approaches. And two of these tools were made in our group. First one is Dereplicator for searching chemical databases, and the next one was VarQuest, which is an improvement for searching not for exact matches, but for variants of compounds present in chemical databases. But since these two tools were like first steps in this area, they are rather primitive, and today's talk is about improving core facility of these two tools with new computational approach. But before I will show what we improved, I will present briefly how Dereplicator works and VarQuest. So both tools start from an experimental spectrum and a chemical database of PNPs. Then it forms so-called PSMs, peptide spectrum matches, and scores how similar this particular spectrum is to this particular chemical compound. After that, we compute statistical significance of each score and report identifications which pass some predefined threshold of their statistical significance. Also, we estimate false discovery rate in our identifications by using traditional proteomics approach, which is called target decoy approach. We simulate false database of false peptides, perform the same steps with them, and estimate false discovery as the ratio of false peptides to the ratio of peptides found in target database. So this ratio is our estimate of FDR. And this, our work, which I present, is about engine of this pipeline. It's about scoring and significance estimation of the scores. But since we are limited on time, I will show only the first part, NPScore. And I will have a poster today where we can talk about significance part in more details. So about score, how we currently score peptide spectrum matches. We need to compare chemical structure and experimental spectrum. We know that molecules in mass spectrometer fragmented, and this fragmented can be simulated, modeled in computer to form a theoretical spectrum of a molecule. And we can compare, since we moved from chemical structure to a theoretical spectrum, we can compare two spectra with each other, and baseline scoring, which was present in Dereplicator and VarQuest software, is simply number of shared peaks between experimental and theoretical spectra. But this model is rather primitive, and it has two important limitations. First, the score is the same for high and low NPS peaks, so it doesn't distinguish between them, but of course it's important. And fragmentation model is rather primitive. It ignores a lot of events which can happen in mass spectrometer instruments. So we developed NPS and NPScore, which first consider peak intensity. We consider different approaches to accounting for intensities, and the best one in our experiment was ranking approach, where we assign ranks for each experimental peak. The largest experimental peak get rank 1, the second get rank 2, and so on. Second, we improved our model by considering additional ion types in theoretical spectra, like water loss, different charges of experimental theoretical peaks, and so on. And for each pair of rank and ion type, we have a different weight. So in basic model, we have only weights equal to 1, and here we have floating points weights for each case. And finally, we consider not only shared peaks, but also missing peaks, both in experimental side and in theoretical side. And the general model is common in proteomics studies for the same topic. And it assumes that there are 3 types of peaks: shared, which occur with some probability which is different for different ranks and type of ions. Also, there are noise peaks, which are present only in experimental spectra, but missing in theoretical model, and vice versa, theoretical peaks, which are present in theoretical model, but missing in experimental. And finally, the score is log odds ratio of probability of seeing this particular experimental spectrum S, in case we have a theoretical spectrum T, to the probability of getting this experimental spectrum out of empty theoretical spectrum. So the higher the score, the more likely we see correct theoretical spectrum to the experimental one. And after some simplifications, which are available in our paper, we finally get something like this, and each adduct is a parameter of NPS. And we learn this parameter statistically from training data. First, we form a training data set of highly reliable PSMs. And then statistically learn each of these parameters, like, for example, probability of getting experimental peak of rank explained by a theoretical ion type ion is just frequency of such events in our theoretical, in our training data set to the ratio of all experimental peaks of rank in the training data set. And finally, we get something like this. So on the left, we see probabilities, which we learned from real data. And when we perform log odds, we move to weights. Different colors means different type of ions. And finally, to the results. First, we made a sanity check. We checked our new approach on linear proteomics dataset, which is well studied, and we know ground truth for it, and compared NPS to baseline Dereplicator and to one of the leading tools in proteomics, MSGF+. And here are the results. You may see green line is on the bottom. It's our baseline, which we are improving in this study. Blue line is our new NPS-based Dereplicator, and red one is one of the leading tools in linear proteomics. And you see that we improve over baseline, but still losing to MSGF+, but it's fine because MSGF+ relies on some assumptions which we cannot consider, like linear structure of peptides and presence only of 20 amino acids in their structure mostly, while we should consider cases when we have 100 different amino acids possible. And since we see that improvements is present, we moved to our target data, PNP data sets, taken from GNPS platform. And here we can't run MSGF+, so we compared only to baseline Dereplicator. And you may see a significant improvement on this data. For example, at strict FDR 1%, we have like 40% improvement over previous method. And finally, we also tried variable dereplication, VarQuest case. Here we took only 3 data sets and also get improvement both in traditional dereplication and variable dereplication, which assumed modification tolerant search. And that is all. I would like to thank my co-authors, Azat and Alexander, and also Hussein, Anton, and Pavel for their help and advices in this research. And thank you to all you for your attention. And I will be happy to answer your questions right here or tonight at poster G09. Thank you.
Thank you very much. We got time for 1, 2 quick questions.
Hi, very nice talk. One question: why do you need the decoy database when you are able to calculate a p-value from the score anyhow? When you have a p-value, you have the null distribution, you can directly have your FDR, right?
Yeah, that's a great question. Thank you. We don't need it for our point of view, but usually our potential users are very familiar with FDR and not very familiar with p-value. So to convince them, we also compute both tools. But yes, you can rely on p-value, but p-value computing is not as straightforward for this task. So we made some assumptions which some people rely on, some people not. So we use both tools.
All right. Thank you very much. In the interest of time, we move on. Thank you.